Hi, everyone. Welcome to the MAS-Seq webinar. My name is Elizabeth, and I'm a product manager at PacBio. With me today is Roger Volden, our bioinformatics scientist. And let me show you the agenda. The talks from today are actually pre-recorded. There are three components. I'm going to give a brief introduction to MAS-Seq for single-cell RNA sequencing. Harsher and Dylan is going to talk about the library preparation workflow using the MAS-Seq kit. Sharon is actually here today live. She'll be answering questions at the end. However, she is sick, so she might not be able to get to all of your questions. We'll do whatever we can. If we are unable to answer any of your questions by the end of this webinar today, we will be answering them over email later. And then thirdly, Roger will be speaking about the bioinformatics analysis. There are some housekeeping items. In the tab of your GoToWebinar, you should be able to see a literature or handout tab where you can download. These are literature resources. All attendees are muted. During any time of the talks and after the talks, you can use the Q&A chat box to ask questions. We will address these questions after the recorded talks. This session is recorded and will be made available after the event. And finally, at the end of the webinar, please take time to fill out the 2-minute survey. We greatly appreciate your feedback. All right, and with that, let's get started with the talks. Welcome to the MAS-Seq webinar. Today, I'm going to give you the introduction to the MAS-Seq for single-cell isoform sequencing. And for the remainder of the talk, I will be off camera. But first, let's talk about what's different for using long-read sequencing compared to short-read sequencing for RNA sequencing. For RNA sequencing, when using short reads, cDNA have to be fragmented to be sequenced by paired and short reads and then assembled computationally. This can only assemble 20 to 40% of transcripts, providing a partial view of isoforms. In contrast, using long-read sequencing, the Iso-seq method, PacBio can offer superior discovery power, giving a complete view of the isoforms. The PacBio Iso-seq method for bulk RNA sequencing is an established application that has been applied to cancer, neurodegenerative diseases, and plant and animal genome annotations. But in the RNA world, single-cell omics is an emerging application. Just like in bulk RNA sequencing, where long reads have the advantage of seeing full-length isoforms, the contrast is even higher in single-cell RNA sequencing. And that is because the single-cell tag information, such as UMI and barcodes, are only at one end of the transcript. With short reads, only partial gene information can be read out. In contrast, using long reads, the full-length isoform plus the single-cell tag information can be read. We know that isoforms are important in discerning diseases. For example, in cancer, the BCL gene has two isoforms. One promotes cancer, while the other suppresses it. PacBio customers have used single-cell Iso-seq to reveal different disease states. For example, applying this to aging Down syndrome brains, they have showed different isoform expressions amongst different cell types. In another recent publication, the authors at Nationwide Children's Hospital showed that the P10 gene in this patient has two distinct mutations in exon 5 and exon 9. They further showed with single-cell Iso-seq that the two variants are expressed in different cell types. Last year, authors from the Broad Institute published a new method called the MAS-Seq method, which is a general amplicon concatenation method for increasing throughput on the PacBio systems. They showed using one SMART cell on the Sequel IIe system, they were able to achieve 33 million reads. not only they were able to recapitulate the same cell types as the matching short read data, but also they were able to identify full-length transcript isoforms. As an example, they showed that the CD45 isoforms, such as RB, RAB, and RO, are expressed in different cell types. We have recently launched the MAS-Seq kit for 10x single-cell 3' kit. It supports the cDNA generated from the 10x chromium systems. From 10x cDNA to library preparation takes 2 days using the MAS-Seq kit. It includes MAS adapters, so no additional library prep is needed. The MAS-Seq kit is compatible with Sequel II, 2E, and Revio systems, and provides an end-to-end SmartLink software solution that outputs gene and isoform count matrix that is compatible with tertiary analysis tools. This ends my introduction. We will now move on to a detailed description of the library workflow and data analysis.
Hello, everyone. My name is Harsharan Dhillon. I'm a scientist at PacBio, and today I'll be going over the MAS-Seq library preparation workflow. And the remaining slides of this presentation, I will turn off my camera. Okay, so this slide walks through the journey of a MAS-Seq user. The MAS-Seq kit is a result of collaboration between 10x Genomics and Broad Institute. The cDNA, which is generated from the 10x chromium single-cell 3' kit, is used as an input into the MAS-Seq kit. This kit is intended for use with the single-cell cDNA generated using the 10x chromium NextGem single-cell 3' kit standard throughput, and an optimal range of between 3,000 to 10,000 target cell recovery is recommended. The cDNA that is generated using the 10x workflow is used as an input into the MAS-Seq kit to generate SmartBells, which are then sequenced on Sequel II or 2E system, followed by data analysis. The MAS-Seq library construction workflow comprises of a total of 8 steps, which can be completed in a duration of 2 days. This slide lists the walkaway time for each step, which comprises of a total of 6 to 6.5 hours on day 1 and 2 hours on day 2. There are several safe stop points throughout the procedure, so if the user wants, they can stop earlier on day 1 and continue with the subsequent steps on day 2. Now I'll go through each step of the workflow in the next slides. Step 1 of the workflow is input cDNA quality control. So the single-cell 3' cDNA generated from 10x workflow needs to be QC'd first by evaluating the size distribution using Bioanalyzer. And typically, the 10x single-cell 3' cDNA samples measured with the Bioanalyzer show a peak around 1 to 1.8 kb. Additionally, concentration of the cDNA sample will be determined using qubit. This protocol supports 2 input amounts, a low input of 15 nanograms and a high input between 60 to 75 nanograms. So if user has cDNA amount between 15 to 59 nanogram, then it's advised to normalize the cDNA sample to 15 nanograms. If a user has a cDNA amount greater than 75 nanograms, then it's advised to normalize the cDNA sample to 75 nanograms. And if a user has a cDNA amount between 60 to 75 nanograms, then normalization is not required. Shown here is an example human cell line GM12878 cDNA sample, which was quantified using high sensitivity DNA kit via Bioanalyzer. It shows a peak of around 1.5 kb. And you can also see that there are some smaller fragments starting from around 500 base pairs, as well as larger fragments up to 4 to 5 kb. The next step after cDNA QC is TSO-PCR. This step uses mass capture primer forward, which is a modified PCR primer, to incorporate a biotin tag into desired cDNA products with the correct structure, meaning that the cDNAs which contain the correct forward and reverse primers will be biotin tagged to distinguish them from the TSO priming artifacts. Step 3 after TSO-PCR is TSO artifact removal, which uses streptavidin-coated mass capture beads to enrich for biotin tagged cDNA products with correct structure, thereby removing the TSO priming artifacts. So some of the best practices for TSO-PCR is first to set up the TSO-PCR reactions on ice. And this is because the PCR polymerase in the PCR mix contains 3' to 5' exonuclease activity, which negatively impacts amplification yield if these reactions are prepared at room temperature. There are 2 different thermocycler programs depending on the input amounts used. 5 cycles are recommended for 15 nanograms input, and 3 cycles are recommended for input between 60 to 75 nanograms. After TSO-PCR, it's recommended to perform cleanup using 1.5x SmartBell cleanup beads, but please note that if the cDNA profile contains smaller fragments, less than 200 base pairs, then it's advised to increase the cleanup ratio to 2x. Best practices for TSO artifact removal step. First is to bring the mass capture beads kit to room temperature. Resuspend the mass capture beads by vortexing before adding them to the tube. And a critical step is when conditioning the mass capture beads with mass bead binding buffer, which can be very viscous. Pipette mix gently with care and avoid generating bubbles by using wide bore pipette tips for mixing. Do not vortex to mix during these steps. And we have noted when excess bubbles are present, lower cDNA recovery will likely occur. Add MAS enzyme to the cDNA sample containing beads, and this is to cleave the captured cDNA products from the mass capture beads. A critical step is to keep the supernatant after treatment with MAS enzyme because it's needed to proceed into the next step of MAS-PCR. But before doing MAS-PCR, perform a cleanup using 1.5x SmartBell cleanup beads. And if smaller fragments, less than 200 base pairs are present, then increase the ratio of cleanup beads to 2x. Step 4 in the procedure is MAS-PCR. This step generates amplified cDNA products containing specific sequences at both ends. So the cDNA sample after TSO artifact removal and cleanup, to the cDNA sample, we add PCR reaction mix and then distribute this into either 16 tubes of a PCR strip tube or into 16 wells of a 96 well plate. To this, the MAS primer pair solutions are added. The pre-mixed primer solutions come packaged in individual tubes, so the users can first transfer the primers into either strip tubes or a 96 well plate, and then use a calibrated multi-channel pipette to add the primers to the samples. The end result from this step is that the amplified cDNA products from one PCR reaction will contain terminal sequences that are complementary to the ends of cDNA products from neighboring PCR reactions. For example, the cDNA products from PCR2 will contain terminal sequences that are complementary to the ends of cDNA products from PCR1 and PCR3. So best practices for doing the MAS-PCR step is first to set up the MAS-PCR reactions on ice, and this is due to the same reason as in TSO-PCR step, since the PCR mix has PCR polymerase containing 3' to 5' exo activity. So please make sure that this reaction is set up on ice. The maximum recommended input into MAS-PCR step is 50 nanograms. A critical step is to make sure to correctly set up all 16 MAS-PCR reactions, because any missing or incorrect MAS primer pairs will result in no or low SMRTbell yield. The default MAS-PCR thermal cycler program uses 9 cycles. And after MAS-PCR step, the 16 reactions are pooled into a single 1.5 ml tube, and a cleanup is performed using 1.5x SMRTbell cleanup beads. Again, if the starting cDNA sample has fragments less than 200 base pairs, then increase the SMRTbell cleanup bead ratio to 2x. Step 5 in the procedure is mass array formation, which enables assembly of cDNA fragments into linear array. So the pooled cDNA sample after MAS-PCR is treated with MAS enzyme, which creates single-stranded overhangs. To this sample, MAS adapters A and Q are added, followed by addition of MAS ligase in order to enable formation of linear arrays. This ligation reaction results in the formation of complete full 16-segment array along with some partial incomplete array molecules that may contain only one MAS adapter at either of the ends or no adapters at all. Best practices for MAS array formation step. The recommended input requirement into this step is 10 micrograms. Critical step is to always add MAS adapters and MAS ligation additive to the sample before adding MAS ligase, and this is to ensure uniform ligation. And I would like to mention that from step 5 onwards, since we have 16-segment arrays, the SMRTbell cleanup ratio is now changed from 1.5x to 1.2x, and also it's recommended to use wide bore pipette tips to avoid accidental shearing or damage of fully formed arrays. So after the MAS array formation, step 6 is DNA damage repair, and step 7 is nucleus treatment. So after MAS array formation, the arrays might contain DNA damage or nick sites shown by these magenta triangles. These are repaired by using DNA repair mix. The incomplete arrays, which contain either one or no MAS adapter, are removed using a nucleus mix. So after step 7, most of the remaining SMRTbell templates should be complete 16-segment array molecules containing MAS adapters at both ends. Best practices for DNA damage repair step. Do not proceed with more than 5 micrograms input into this step. Adding more than 5 microgram into the step will likely result in incomplete digestion of partial SMRTbell template arrays, resulting in low P1 loading performance during sequencing. Make sure to pipette mix with wide bore pipette tips. This is to ensure to minimize DNA damage or DNA shearing to the full-length SMRTbell template arrays. After DNA damage repair step, perform cleanup using 1.2x SMRTbell cleanup beads using wide bore pipette tips, and then proceed with step 7, which is the nucleus treatment step. In the nucleus treatment step, nucleus mix is added and the sample is incubated for 1 hour. After nucleus treatment, final cleanup is performed using SMRTbell cleanup beads, and then the library QC is performed by determining DNA sizing using femtopulse system. So the expected final library size should be around 10 to 15 kb. If the users don't have access to a femtopulse system, they can perform DNA sizing QC using a bioanalyzer system. But please note that for bioanalyzer, the electropherogram profile of the final library may partially overlap with the 17 kb upper marker. And DNA concentration QC is performed using Qubit dsDNA high sensitivity assay kit. The typical final SMRTbell library yield, starting with 5 microgram input into DDR step, is around 10 to 25%. A troubleshooting tip is if the SMRTbell library yield is higher than expected, so if the yield is greater than 35%, verify the size of the library via femtopulse. If library has a shoulder of smaller insert sizes, it might be that the library was incompletely digested. So in that case, consider repeating the nuclease treatment step for another hour before sequencing the final library. This is example femtopulse trace of final SMRTbell library, where blue trace represents the sample post-mass array formation, and black trace represents final SMRTbell post-nuclease treatment. You can see that there is a significant shoulder of smaller fragments less than 10 kb shown in blue trace, which corresponds mostly to incomplete SMRTbell arrays, which are significantly removed in the trace post-nuclease treatment. Step 9 is sequencing. So in this step, we prepare the final SMRTbell library for sequencing via ABC. ABC stands for annealing, binding, and cleanup. So in this step, a sequencing primer shown in black is annealed, followed by binding the polymerase and cleaning up the complex. This final cleaned up complex is then ready to be loaded onto Sequel II or IIe system, where 30-hour movies are collected, followed by auto-analysis using the SMRTlink software version 11.1. This slide shows example MAS-Seq sequencing performance for a human MAS-Seq library. The electropherogram on left shows the final library QC, which has a mean insert size of 14.2 kb. The library insert size is consistent with the mean hi-fi read length distribution plot shown on the right, which shows a major peak at about 15 to 16 kb. The table on the right shows hi-fi metrics. So in this case, we got 2.2 million hi-fi reads with 34.5 GB hi-fi yield and a median hi-fi read quality of Q31. And with that, I will hand over the presentation to Roger, who will go over the analysis.
Hi, everyone. I'm Roger. I'm a bioinformatics scientist here at PacBio. And today I'm going to talk about read segmentation and the single-cell Iso-seq workflow. For the duration of the presentation, I'm going to have my camera off to focus on the slides. The MAS-Seq bioinformatics workflow goes from hi-fi reads to segmented reads and ultimately into an isoform matrix for single-cell tertiary analysis. In the SMRTlink encapsulation, this is going to be two separate workflows, being read segmentation and single-cell Iso-seq. To kick this off, we go from hi-fi reads to read segments using a tool called Skira. After this, we do primer, UMI, and barcode extraction using Lima, Tag, and Refine. After this, we do barcode correction and UMI deduplication using Correct and Group Dedupe, which will ultimately give us our unique molecules. We take all of our unique molecules and map them to our reference genome. Thereafter, we use collapse in order to get our unique isoforms for this sample. Those isoforms are then structurally classified in our SCONTI3 replacement pigeon, and this will also make a tertiary software compatible gene and isoform matrix for use with Seurat, ScanPy, and Kana. I'm now going to go into more detail on each of the individual tools that I've laid out here. The first tool in our pipeline is called Scira, which deconcatenates reads into constituent cDNA molecules. On the figure to the right, you can see we have a length 4 array with adapters A through E separating them. What Scira will do is it will search for these A through E adapters and extract the constituent cDNA molecules out of them. And each of these cDNA molecules represents all of the cDNA molecules that were produced in your 10x workflow. The command for this just takes your hi-fi BAM file as well as your MAS primers, and it will produce a segmented BAM file. Next up, we have Lima, which will remove your primer sequences. So as you can see in the figure to the right, this will remove the 5' TSO as well as the 3' 10x primer. And what you're left with is your transcript, which contains your poly A, your UMI, and your cell barcode. All this requires is the option for Iso-seq and the 10x primers, along with your segmented BAM file. Following Lima, we extract barcodes and UMIs using Iso-seq 3 tagged. What this will do is it will shunt your UMI and cell barcode into their own BAM tags, and this is used later for barcode correction. Doing this requires you to input a design for your transcript, and this is reading 5:3, so you tell it that you have a transcript with a 12 base pair UMI and a 16 base pair barcode, and this will search for those sequences and put them into your BAM tags. Next, we have Refine, which removes poly A tails and artificial concatenators. As you can see in the figure, all we're doing is removing the poly A tail from the 3' end of the sequence, and this uses the option require poly A and searches for concatenators using your 10x cDNA primers. So this would not capture under deconcatenation with Scira. Next, we use Iso-seq 3 correct to fix cell barcode mistakes. This takes an include list that we take from 10x as well as our reads to correct cell barcodes that are within a certain edit distance of canonical barcodes. It should be noted that the cell barcode include list that is provided directly from 10x is oriented from 3 to 5', which is why we reverse complement all of our sequences here. Following this, we also use SAM tools to sort the output BAM file by the cell barcode and UMI. Following the sorting, we can use Iso-seq 3 group dedupe to deduplicate molecules based off of their UMIs. So this is just going to look for your unique molecular identifiers in each of your reads, check that the barcode for the cell that it came from is the same as the other reads that match that UMI, and we'll deduplicate them from there. We take our deduplicated molecules and align them to our reference genome using PBMM2, which is PacBio's wrapper around Minimap2. This will produce a set of spliced alignments that we can later collapse into unique isoforms. Iso-seq 3 collapse produces unique isoforms based off of their transcript features. So this will look at your transcription start and end sites, as well as all of your splice junctions, and collapse them into unique isoforms based off of those features. This will produce a collapsed GFF file, which can then be sorted using our next tool, Pigeon, to create a sorted GFF file. Following collapse, we can use Pigeon, which is our Sconti 3 replacement, to classify isoforms into their structural categories. For those of you familiar with transcriptomics, you're probably also familiar with this classification system, which will tell you whether your isoforms match all of your splice sites to result in a full splice match, or maybe you're missing terminal exons, which are incomplete splice matches, or if you have novel isoforms. This requires a smattering of different reference files. The ones that we are particularly interested in are the abundance of each molecule, a list of poly-A motifs, as well as a bed file of cage peak positions to determine how reliable our 5' and 3' ends are. And this will produce a tab delimited classification file. Following pigeon classification, we can also filter out isoforms that are likely to be artifacts. So isoforms that align to intronic regions of the genome, as well as reads that span both intronic and exonic regions will get filtered out. And this will produce another tab delimited classification file, just filtered. Following filtering, Pigeon will also produce an isoform matrix for tertiary analyses. This will take your filtered isoforms, produce your barcodes, genes, and matrix file, as you would see with 10x Genomics Cell Ranger software, and this will be able to produce cell type clusters in your favorite tertiary analysis suite, such as Syrah, ScanPy, or Kana. And with that, I'll be happy to take any questions.
All right. We have a lot of questions. So what I'll do is, I think what we can do is so we've answered a lot of the library preparation questions, but I think it would be helpful for me to just verbally say some of them. And while I do that, Roger, if you can look at some of the bioinformatics questions that are still streaming in. So let me go through the library questions first. I think the biggest question people had, besides the technical details, was that does MAS-Seq kit that we just released support anything that isn't the official use of 10x 3' cDNA? So the questions were such as, does it support the 10x 5' kit? Does it support bulk Iso-seq? Are there plans to support other amplicons? So let me just say the MAS-Seq for 10x single cell 3' kit that we have just released and is available for order now officially supports cDNA generated using the 10x Chromium 3' kit version 3.1. It does not support the 5' kit due to the handles for the 3' kit and the 5' kit are different. So the TSO depletion step would use different oligos, and the one that's included in the kit right now supports the 3'. And therefore, you would not be able to successfully deplete the TSO artifacts that are in the 5' cDNA library. It does not support bulk Iso-seq. The handles are completely different there. We do know that our customers are very eager to apply the MAS-Seq concatenation method to all your other favorite amplicons, and rest assured that is what we're going to work on supporting you next. But right now, this kit supports cDNA from the 10x 3' kit. I think there are some technical details on asking what TSO is, how this artifact arises. So TSO, I'm sorry we did not spell out the full word. It's template switching oligos. The artifact has a phenotype where the TSO appears on both ends of the molecules as a mispriming during the first strand synthesis. As a result, these reads are not considered full length, and our goal using long read sequencing is to get full length information from the 5' and the 3' end. So we remove these TSO artifacts. There was another technical question on why during the nucleus treatment step, we're doing 1 hour. Some of you are familiar with the HiFi workload, that's 15 minutes. So during development, we tested different nucleus treatment times. 1 hour resulted in the most optimal yield of full arrays, which gives us the most number of reads. Let me take a look at if there's other library prep questions. Yes, I think we can get to the analysis, and maybe I'll answer some really high-level analysis questions first. They were asking, is this included in SmartLink? What does it support? So after Roger's presentation just now, I want to emphasize that, yes, this is supported in SmartLink. using the read segmentation and single-cell Iso-seq analysis workflow. You don't need anything else. It will output at the very end a gene and isoform count matrix. And that matrix has the standard format of, matrix.mtx, genes.tsv, barcodes.tsv, so you can take them into Surat, SCOMP, and Kana. And the way we're doing classification is using Pigeon. So those of you who are familiar with a tool called SCOMP, Pigeon is the PacBio implementation of SCOMP3. We did, however, take some of the features out of SCOMP3. Those of you who use SCOMP3 is familiar, it has lots of amazing features for classifying transcripts against gen code annotations. We extracted the core features of SCOMP3 to implement into Pigeon so that we actually can classify not just known transcripts against gen code. We also output novel isoforms of known genes and also novel genes. However, in the default output matrix, we only output isoforms of known genes. It could be novel isoforms and it could be known isoforms. We don't output by default novel genes since they won't even have a gene name. It would have something like novel gene 123. In the command line, you will be able to additionally extract these novel genes. And by default, we are only supporting human and mouse due to the need to support comprehensively the correct genome and annotation GTF format. However, if you have non-human and now mouse samples, you can still run, and we recommend you still run the read segmentation and single-cell Iso-seq workflow, which will complete up until the point of mapping to the genome, which you see Roger's talk is pretty low down the pipeline. Once you have reached the level of UMID duplication, so you have the unique molecules, you can take those data out and then work on the command line for your genome and your annotation. And that can be done on the command line. If you want to check out the URL that kind of shows all of this tutorial, isoseq.how is our biggest command line repository where we have examples and toy tutorials that you can download. So I think I've covered the general informatics questions now. We have a lot of questions in the chat. So Roger, some people are having issues with hearing me, but Roger, do you have issue hearing me? Okay. Yeah, I'm sorry. I'm not sure why there are some audio issues, but this session is recorded. If you are unable to hear me, we will be sending you the recording after the session. Let me go through, so maybe let's do this, Roger. I'll go through some of the questions. I will say them, and if you can answer them. Let's see. Is the analysis pipeline available for downloading? Yes, we've already answered this question. Yes, you can download it from our official software. Let's see, what else? Are there any data sets available? Yes, we will be upgrading our data set. So we actually had a minor bugs that we're fixing this week, and we will be updating our PBMC public data sets by the end of this week. I think there's a question, Roger, on looking at, we talked about pigeon filtering. So they're asking, why exclude reads that map to an intron and an exon? Could this be possibly missing an exonization events?
Yeah, we would be missing out on those events, but in most of the cases that we've seen, these are mostly artifacts.
Yeah. Okay, here's another question. This one I can answer. Are there any differences between the Sequel 2 and Revio on 24 hours? This is a great question. We will be releasing in the near future what the MAS-Seq kit performance looks like on Revio. So I think we talked about 40 million reads. That is only for the Sequel 2 and 2E systems. We know on Revio we will be getting a lot more than that. We'll be updating what that performance looks like on Revio very shortly. Yeah, so here's a lovely question. Well, Roger, currently, are we supporting fusion gene identification?
This is something that we're actively working on.
Yeah. Okay, a question on, so yeah, some attendees are familiar with my old GitHub script called Cupcake and asking, you know, do we still need Cupcake? How's it integrated? So yes, as of the release of SmartLink 11.1 and the Biocana command lines on iso-seq.how, I will be deprecating Cupcake as almost all of the features have been replaced by our official software. It is faster, it is more scalable, and it integrates with our official pipeline. So if you are a Cupcake user, thank you for using it. Please migrate to our command line or our SmartLink via iso-seq.how or visiting our website, pacb.com/software. Okay, this is a great question. Is gene quantification and differential gene expression analysis possible with this data?
This isn't something that we have explicitly tried, but I think this should be possible.
Yeah, and I want to mention that we are not supporting those tools right now, but we actually already had customers who were working on single cell differential analysis, several publications, and in fact, there's multiple tools. ScissorWhiz is one that comes to mind. We will be compiling some of these customer tools shortly, and I think what we'll do is put them on iso-seq.how as well. Here's another question. Does SmartTool take FOSQ as input data downloaded from Geo? Yeah, I see what they're saying. It's like if they already have a, you know, like a an uploaded publicized data set from Geo, would they take FOSQ files? Yeah, I can answer this. So no, in general, the majority of our tools work with band files that have the correct tag information. For example, we store the UMI and barcodes in our band tags. I think there are some more downstream processes, such as if you already have the deduplicated FOSQ files, you could be mapping them to the genome without the need of a BAM tag. But generally, if you look at our pipeline, it is very BAM-centric. Okay, and I think there's a follow-up question on, I think it's a continuation of the question on the pigeon filtering. Some genomic regions could be annotated as introns in some GenCode transcripts, but as exons in others. How are they treated by pigeon? I can answer this question, so because we are using the Sconti classification scheme. So in general, when we see multiple options of matching against a GenCode annotation, we prioritize those that will qualify them as matching an exonic transcript. So let's say if there are two entries in GenCode, one says this is a pseudogene or it's going to make it antisense, and the other one's going to make it matching a perfect coding transcript, just hypothetically. Pigeon or Sconti will prioritize the one that designated as a coding transcript. So there is like a ranking order. So yes, if there's a GenCode says this might be a coding, we'll definitely prioritize it to say it's probably coding. Okay, yeah, there's another question. I think this is more for me. Can we sequence the MAS library with more smart cells to get higher coverage? Absolutely, and you know the MAS-Seq library that with the kit that we generate right now can easily run up to 2 smart cells. In some cases, we've been able to run 4 smart cells in SQL 2 and 2E. However, again, we will be looking at this performance on Revio with the increased throughput on Revio. We're quite confident that you will not need more than 1 smart cell. Here's an question on difficulty to work with any animal models. I think this is thinking about more like analysis. So there's really no difference in analyzing this against human or mouse. The reason we only support human and mouse is because we have very well annotated GenCode models. And actually, if you look at the SmartLink report, we actually also include cage peak data and poly-A motif that are commonly seen in eukaryotes. These are all something you can supply when you run the command line version of Pigeon. It's just that, you know, when we do the official development, we don't really have alternative animal models to test on. It's also difficult to make sure they support, you know, for example, you know, cuttlefish or octopus. So yes, if you have your animal models, you have a GenCode like GTF annotation, definitely you can use it with Pigeon. The cage peak data and the poly-A motifs are completely optional. The only thing that's really required to run Pigeon and get all of the subsequent analysis is you must have a reference genome and you must have a annotation in the proper GTF format. What are the longest isoforms we can sequence with MAS-Seq? I think this is a two-way question because it depends on the maximum cDNA length that we're seeing from the input cDNA. And right now, some of the constraint might be coming from what is the longest cDNA that we can generate from the upstream 10x chromium system. And also the other is we are doing 16-fold concatenation. And if you have, let's say we're not using the 10x chromium, we're getting very long cDNAs, and if that were compatible with the MAS-Seq, one of the considerations is would we have trouble concatenating the really long transcripts? And I think I'm going to combine that with Castle's question on, you know, when are we going to support MAS-Seq for bulk? Again, this is something we are really prioritizing. There are multiple technical factors we have to consider, such as with bulk Iso-seq, using the Iso-seq Express kit, we see really long cDNAs, right? Up to 10, sometimes 15 kilobases. We want to ensure that we're able to still, you know, capture these long transcripts, provide a good yield increase with the concatenation, but also giving you all the transcripts that are in the original sample. Let's see. I think these are most of the questions. I think there's some questions about quantitative and being able to see differential. Again, I think we are not currently supporting software for doing differential analysis, but we absolutely have customers who, even before MAS-Seq was available, have done differential isoform expression analysis using their software. So I'll be creating a resource pointing you to those software soon. Okay, yeah, I'll answer the next two questions I see. Is this kit only for poly-A RNA because of the kit? Yes, so yeah, we're constrained. We get the cDNA that comes out of the 10x chromium system, which does a DT priming. So yes, only poly-A RNA will be captured. Next question is how many samples are appropriate to run on a SMRT Cell for MAS-Seq? Can we multiplex? So if you look at the library workflow, it already generates a library with the MAS adapters, and the adapters currently do not offer multiplexing solutions. So as of now, MAS-Seq kit, one reaction, one library is for one sample only. Okay. All right, I'll wait just a tad more. So comments from one of the attendees, you know, with the Revio throughput, there will be a desire to multiplex. Yes, we are absolutely taking that comment into consideration. All right, so thank you for staying. When you leave the webinar, we would greatly appreciate if you can take 2 minutes out of your time to fill out the survey. Having your feedback really helps us with improving our webinars. Okay, I will answer the next question. Is there a website that explains the individual steps of the analysis workflow? Yes, so in addition to the slide deck and this webinar, which you will receive shortly, you can also go to iso-seq.how. Hopefully that's a very short URL now. We explain in detail, as much as we can, the workflow. And again, if anything's not clear on iso-seq.how, please let us know. We will update the documentation accordingly. All right, I think that's all our questions. Thank you for joining the webinar. I'll talk to you soon.